Hi, this is Chris Kiefer, and welcome to episode 50 of the Power of ERDC podcast. Today, we're talking with an ERDC researcher who examined something ancient and discovered something novel. Our guest today is Dr. Robyn Barbato, a senior research microbiologist and the leader of the soil microbiology team at ERDC's Cold Regions Research and Engineering Laboratory, or CREL. Barbato and her team recently discovered 26 previously unknown species of microorganisms frozen in permafrost that was approximately 40,000 years old. We will talk with her about this discovery and about how these tiny organisms have the potential to support U.S. military operations in extreme cold regions. Robyn, hey, thanks for joining us today. It's great to have you back on the podcast.
Thanks so much, Chris. It's great to be here.
Yes, absolutely. So I want to get into the story of your latest discovery, but first, just to kind of explain it to the audience, can you talk about studying microorganisms and how studying microorganisms and finding these new species can help the U.S. Army?
Yes, absolutely. And first, I want to say thank you so much for the opportunity for us to talk about our really awesome research with you. So biotechnology is a priority for the Army to create advanced materials for biomanufacturing, which is manufacturing biological things, and improving soldier performance through things like probiotics, like the yogurt that you might eat. So the Army, it's really important. They need a resilient supply chain, one that gives them what they need wherever they are. And cold regions pose really unique challenges in procurement and delivery of these needed supplies because it's long distance, or maybe there's no infrastructure for roads, maybe it's extreme weather conditions. So this point of need for biomanufacturing is really, really important. And also for sensing the environment. So something interesting about microorganisms is you can't see them, but they're nearly everywhere. And they have really interesting capabilities, like they could replicate really quickly. For instance, Escherichia coli, which is one of the most studied bacteria on the planet, it can replicate every 20 minutes. So think of every 20 minutes, it's doubling. So it grows more and more and more. And so these organisms in the environment, they build things and they break things down. So Werner Suhm said microbes are the best chemists. So these unique properties that they have really make it advantageous to the Army, because the Army is pressed with these particular needs like I just outlined. So maybe the Air Force has to de-ice planes. Maybe the Navy wants to reduce transit times and meet these logistical demands. So we can take some of the technology, the biotechnology, the things that these microorganisms are doing, and apply it to these military problems.
Sure, and you mentioned logistical challenge in, a far-flung region, a region that's difficult to get into, like the Arctic. The advantage, I guess, is that they're already there, and they're there in really large numbers. The microorganisms,
Yes,
you're
yes.
right.
Yep. And
Yeah.
oh, sorry.
Go ahead.
Don't tell NASA, but there's more organisms on the planet, more bacteria than stars in the sky. So they're nearly everywhere. You just can't see them.
Yeah, sure. What would be some examples, high-level examples of the sorts of applications, technologies, ways that these microorganisms can be used?
So one thing is they can be used for synthetic biology. So synthetic biology is engineering bacteria or other microorganisms to give them new capabilities. Bacteria are really fascinating because they can exchange DNA, and there's three main ways that they can do it. Um, and one of those ways is just taking up DNA that's around them. Well, if that DNA has a really interesting gene, like for antibiotic resistance, they might develop that new capability. So, with that in mind, of they can do that in nature, but we can also do that in the laboratory. A good example of synthetic biology that you might be familiar with is diabetics take insulin, and they used to harvest that insulin from cows and pigs, but now there's a bacterial factory. So a bacterium called Escherichia coli that I mentioned before, it's actually engineered to produce insulin. So this adding genetic capabilities is one way that microorganisms could be used for various applications. There's also ways that they can make antifreeze, and that's something that we studied for this effort.
Yeah.
So if you've ever been to the airport in the winter or somewhere really cold, they have to de-ice planes on the tarmac about every 30 minutes. They use ethylene glycol, which is toxic if ingested, or propylene glycol that has a lower toxicity, but it's banned in certain countries. So um, bacteria make antifreeze because they're trying to survive in these frozen environments. So one way that we can use this for applications is for de-icing planes. The other thing is in the military between 2024 and 2025, the U.S. Armed Forces, there was a 42% increase in cold weather injury rates in that time alone. So new ways to counter frostbite. Imagine being outside where it's cold, and if there was some sort of cream, well, if there's an antifreeze protein from a bacteria that we could add to a cream, then maybe war fighters could be out in the cold longer. I mentioned before too how microbes are the best chemists. They can break down waste on forward operating bases. So that's another effort where we're looking into how they can degrade waste products that the military is using when they're in certain locations. Basically, how much time do you have, Chris? Because I could go on and on.
No, that's great, and I think that's what makes it such a cool topic is, I mean, I would imagine these are things that the average person does not think about that often. I mean, it's almost kind of mind-blowing when you think about all the capabilities from something that we can't even see, but as you said, is more abundant than the stars. I mean, it's amazing, so it's really cool. Yeah,
Yeah.
I know, it's exciting for you. T Tell us about this latest discovery. Was it 26 totally undiscovered species, in 40,000-year-old ice? How did it all come about?
So we're funded by the Defense Advanced Research Projects Agency, and their program's called Ice Control for Cold Environments. And our goal was to find new biotechnologies. So find new bacteria that could combat ice or modulate ice. So that could mean preventing ice formation or ice recrystallization. That's the antifreeze I was talking about. But also, bacteria can nucleate ice. And if you've ever gone skiing and you see blowing snow, that's actually a bacterium that nucleated that snow that they're putting on the hill. So,
Huh.
in some cases, for mobility in cold regions, you actually want it to stay cold. Think about if you're driving on a lake, you want that lake to stay frozen. It's much
Sure.
easier to drive on a frozen lake than drive on a melted lake. So these are the
Yeah.
types of things you're thinking about. And permafrost is really interesting and unique because it's ground that's been frozen. So these tiny bacteria have been surviving in this frozen environment. And so we thought gosh, they have to have these kinds of proteins that we're interested in. Um, and so we characterize 50 bacteria, really characterize them really, really well for DARPA. And just over half of them are new species. So that means they haven't been discovered previous to us discovering them.
Yeah, and so, I guess to define for our audience, permafrost is what, ground that's been frozen for at least 2 years? Is that kind of the correct definition of what permafrost is?
That's it, Chris. Yeah, you got it.
Good deal, and it's something
And
that
it could be longer. It could be longer than
right.
that too. Yeah, yeah.
Like, in this case,
So
and obviously, it's something I was gonna say you studied for a long time. In fact, people who are interested in this topic can go back. You've been a guest on this podcast before. I think we were looking at yesterday was maybe episode 13. So if you scroll way back, you can listen to Robyn, talking more about the research she does. But this particular discovery in the scope of kind of how long you've been doing this, I mean, what was unique about this, finding new species? I'm guessing that doesn't happen that often.
yeah, so microorganisms, we have recipes for how we can grow them, and only 1% have been cultured in so far as science, right? So that means, what does culture mean? Culture means we've taken them from nature and into the laboratory. And in order to do that, we grow them using specific recipes. And an example that might you might relate to is if you've ever gone to the doctor for strep throat, they'll stick a swab into your throat and then they put it on a petri plate. We do a similar sort of thing, except we're not putting it on people's throats. We're taking it from the permafrost and then we're making recipes for the organisms to grow. And when you use the same recipes, you tend to get the same kinds of organisms because of the wide diversity of microorganisms in nature. However, the permafrost is really unique, especially the permafrost in the Krell permafrost tunnel, because it spans a long range of ages from about 11,500 years ago to about 40,000 years ago. And so it's almost like a slice through time. So we happen to have really unique samples that we are isolating the organisms. And that's, I think, how we discovered these new species, because they were kind of suspended in this frozen quiescence for a really long time. And they're active. Some of them are active in permafrost. Some of them are just waiting to until the conditions are right. But I think that's how we stumbled across something quite interesting with regard to new organisms for biotechnologies, these novel species.
Yeah, tell us about when you first knew that you had discovered something new, just kind of what that was like. Because I know there was this kind of a delay. You've got to get it back to the laboratory and revive it and do all the things. I mean, kind of when did you know the richness of the discovery you had made, and kind of what went through your mind when that happened?
That's a really great question. So like you said, it takes quite a bit of time because we not only sample from the tunnel, we sample from a variety of Arctic locations, and we need to get the sample back frozen, because if it's not frozen, then it's not representing where we just took it from. And so there's time and energy to take it, sample it, then bring it back, and then in the laboratory, it might sit in our freezer for a little while before we take it out and we're start isolating different microorganisms. But when we found out that these are novel species, I just got so excited because um, even though there's such, 99% of organisms haven't been cultured, there's still challenges with regard to collecting samples, isolating in the laboratory. There's more bacteria on you than your own human cells, so you really have to be super careful. in isolating because you want to make sure that this truly is from the environment that you said it was, like the permafrost. And so that's really exciting. And we use DNA technology to help to identify what the organism is. And that's when we know if it's a novel species. So we basically take the DNA out of the organism and then we run it through a pipeline and we match it to known databases. And if there isn't quite a match, then we understand one, it's novel, and two, it's how related it is to a neighbor. And I think that piece is really exciting. And I think we've been doing this for so long that I didn't really realize how exciting it was until other people started telling me how exciting it was, because so many times, Chris, we're like, oh, this organism is so cool, interesting, came from permafrost. Oh, we already have it. Okay, let's do it again. Oh, we already have it. So to have these 26 that are new, it not only is really exciting because we're contributing to new knowledge, but also it shows how many other new ones there might be out there. So someone listening right now might think of a really unique environment that people don't go to that much or isolate from, and have an idea of this could be a really neat place to try to isolate for biotechnology. And I think that's really exciting. And that it happens in a team, so that we have field experts who go get the samples, we have experts who isolate the organisms, we have experts in what's called bioinformatics who are looking at the DNA and matching it to databases. So it's then that we come together and say, okay, we have something really cool here. And so, yeah.
Yeah, I saw a quote from you, maybe in the press release about this. I think you called it a new biological frontier, microbiological frontier or something. I mean, do you kind of, I mean, really kind of feel like you're on the cutting edge of something, brand new exploration?
I do. And you know, microbiology is not new, right? We've known about permafrost, or that's why Krell came to be, right? Is because they're trying to build the Alaska Canada Highway, and they're like, "What is this frozen stuff? It's really difficult to engineer a road or infrastructure in this area." So those things aren't necessarily new, but as our technology advances and thinking about biotechnology and using new organisms rather than the same old ones that, yeah, they're really well characterized, but what about all the other ones out there that can be used? And that's why I think we are in a kind of microbiology frontier to discover these new things and also show the world that it can work. Because Chris, you mentioned, oh, I didn't really realize that something biotechnology, that I was using it or that it's available. I think those examples, there's so many examples, and we can make way more. Again, because of the fascinating processes of microorganisms, that they have really unique genetic capability and can break things down or make proteins that are advantageous. So
Yeah, I realize this question I'm about to ask, maybe you don't fully know the answer because I know you're still probably studying these new species, but how different are they from previously known species? Are they, kind of similar relatives, or are they kind of dramatically different?
I would say they're similar to their relatives. We know the genus, but the species is unique. And just to
Mhm.
give a contemporary example again is, where Homo sapiens. So Homo is our genus and sapiens is our species. And then there's Homo erectus and other species of humans. So imagine that, but in the bacterial or microbiological context. So
Yeah.
it's like if there was a new species of humans, we're like, whoa, that's pretty wild. It's more common in bacteria, but it gives you a sense of it's not it's like something quite different. Now, as we better characterize them and understand them, we'll get a better sense of what they're similar to with regards to what they can do. We know we have a good handle on the genetic component, what they potentially can do, but we're still in the laboratory testing them and poking them and seeing what they actually do.
So that might preempt it. I was going to ask you next on what sort of potential that these technologies can offer, and maybe what you're telling me is that is maybe too early to say. I mean, not necessarily new species can offer.
Oh, that needs to be, yeah, I think it's early to say, but what we do know with the research that we're doing right now is that we have novel bacteria that we can genetically engineer for synthetic biology. We, in collaboration with our partners at the Naval Research Lab, we learned that we can get them to glow green, we can get them to glow yellow, we can insert other genes into them to have them make more antifreeze proteins. And we submitted a patent on that effort. We also know that they can break down different polymers that's of interest to the Air Force, like coatings on. So sometimes microorganisms colonize coatings and they can break them down. So we have some novel Arctic species that are that can break down those polymers, and so better understanding how they use those polymers so that we can combat when they're on military infrastructure and vehicles. So that's another area. So we have some examples, but there's so much more that we don't know that we're researching.
Sure, sure, sure. And that's what's exciting, I guess, too, is to see what is to come yet. Um,
Yeah, yeah.
cool. Yeah. Just, I think to kind of help emphasize both how important this effort is to the Department of War and even kind of how complicated it is. You sit on a Department of War panel that examines how to best harness biotechnology. Can you talk a little bit more about that?
Yeah, so I'm part of Department of War Community of Interest on biotechnology. And I'm also the co-chair of the Tri-Service Microbiome Consortium. So that's a consortium of microbiologists across the services, Army, Navy, and Air Force. And we are, especially through the biotechnology community of interest, we are trying to expand on capabilities to do biotechnology, scaling up from my tiny test tube actually to making enough of it where you can spray it on a plane to de-ice it. So we're discussing that, and one of the elements of that is to make sure that we're doing it properly, that we have all we're considering all different things of where we harvested the organisms, how we're using it. So we keep all of those in mind and have those discussions. And the discussions span from a warfighter performance to sensing in the environment to biomanufacturing. And it's really exciting to come together on this new frontier to help study this, but also how to show that these can really be helpful in theater.
Yeah, sure. And when you look at some of the potential that these species and microorganisms in general offer, is some of the I mean, is the focus on like the specific biotechnologies that can be developed from this applications from these organisms themselves, or is it on studying these microorganisms that have survived in extreme cold for so long and maybe learning from what they've done? Is it a combination of both?
Chris, it's totally both.
Hmm.
Because you know for the our goal is to develop technologies for the army, so that's driving what we're looking at, like this question about ice modulation of um inhibiting ice in certain circumstances, or making ice or snow in other circumstances. And so that application is always the driving force of what we're doing, because we're interested in transitioning the technology. But then when you step back, I just got the chills, because, you know, when I'm in the field, I'm holding 40,000-year-old permafrost, this capsule of history, right? And that the microorganisms either are surviving or slowly replicating over that huge period of time that they've been frozen. And so there's just fundamental research questions surrounding that. Like, can we isolate anything? Could we get anything out of it? What kind of capabilities do they have? Is it reflective of some different environment? Because thinking about, at least when you first enter the tunnel, is there's like a bison horn sticking out the wall, or a mammoth femur, and you're like, wow, this was in a totally different age than I'm living in right now. So I think that fundamental element to it and discovery is also really exciting. And thinking about how for thousands of years they've been living under these conditions, right? And so what kind of genes do they have, and what kind of proteins? I mean, they are in a stressed environment. So it takes a lot of energy to maintain the genetic material to make proteins. So it's very strategic on how they survive and what you see as the outcome of these different stressors. So, it's not only a cold environment; it's a super salty environment in these permafrost pores. So, just thinking about how they've adapted over this long period of time is super exciting to me, and thinking of how they're living in these tiny spaces and surviving.
Yeah, sure. And that's probably a good segue we ought to talk about that we haven't really gotten into yet is the permafrost tunnel itself. Maybe if you could both give just a quick high level explainer for people that aren't even familiar what it is. That's where you found these samples, and maybe even talking about what makes it such an important research for the Army.
Oh, yes. Oh my gosh. The permafrost research tunnel is about 20 minutes north of Fort Wainwright, and we have at ERDC the Alaska Research Office on Fort Wainwright. So it's a resource that they are working in constantly, and the tunnel is a tunnel into the permafrost. And it's not only permafrost, but it's terrestrial ice features like ice wedges and thermokarst. And in the recent years, it's significantly expanded. When I first came to Krell, it was like one corridor and a little split off. Now you could get lost. It's like, make your second ride. And it's just expanded tremendously, which has also expanded the underground terrain, where you can see the heterogeneity in the features, and imagine, wow, this is really important for mobility, because if I'm looking at this ice wedge that's taller than me, and I'm really tall, Chris, so it's taller than me, and it melts, then how that's going to change the surface landscape. So it's significantly expanded. It's also owned by the Army, so that's a really important part of this, is that we have unique access to it. We can sample from it. We can study it through such a variety of ways. And then we have researchers, like I said, at the Alaska Research Office, where they just have so much fundamental knowledge about it. Constantly, I go there and I think, I want this and this, and I think it's here and there. And they said, Robyn, actually, you want this and this based on what you're describing, and you should go here and over there.
Yeah.
And these folks are like Dr. Tom Douglas, Dr. Amanda Barker, Mr. Dragos Voss. They can help. They just know so much about above the tunnel, but also into the tunnel. So that institutional knowledge is incredibly critical to make the right decisions for executing really good research.
Yeah, sure. And staying on the topic of facilities, I also kind of want to talk about, so when you all find these 26 new species, you've actually got a library, where you can put these and other microorganisms. Some people have libraries of books. You all have a library of microorganisms. I'm going to need to read the name. The Innovative, Collaborative, Exploratory Cold Regions Organism Library for Discovery and Biotechnology. Much easier to call it ice cold for short. But tell us a little bit about that library and the potential that it offers for you all.
Great. Yeah, thank you so much. So it's basically a collection of bacteria and fungi from cold regions. So cold regions could mean the Arctic, Antarctic, mountainous regions. The atmosphere is pretty cold, wintertime conditions, snow, glaciers, terrestrial ice, permafrost. I mean, just a lot of
Yeah.
different things. Yeah. And so in our library, we have nearly 250 isolates. And then for the DARPA ICE program in particular, we characterize 50 in tremendous detail, and that's where we have the 26 novel ones. I created it because for two reasons. One, for the microbial surveys that we were doing by collecting permafrost and then looking at the genetic material itself, there was just so much that didn't match to any database. I was like, what is this? What we need to know what's in here. And we just didn't have good information in the database. And that is ever expanding. So it was for that reason, but then and the predominant reason is that we can have isolates for biotechnology. So you know, these unique organisms from cold regions, there's very limited collections specifically for ones that grow in the cold. And when I went to the American type culture collection or the German culture collection, I wanted some cold ones, and they were listed on their website, but they didn't actually have them. And so I was like, we need to start doing this. So it's a very humble attempt. It's nothing, nothing tremendous, but it's our effort to have these organisms available that we know quite a lot about, and then we can collaborate with other people. So if someone has interest in just understanding how organisms can grow at low temperatures, right? They bacteria have been found to grow as low as like -15 or -20 degrees Celsius, but maybe, we have one in our library that can grow even colder. It's an opportunity to collaborate. And Chris, they're just beautiful when you spread them on a plate and you can see they have different colors. They have unique capabilities, their metabolic potential, what kinds of carbon they can utilize, and what kind of contaminants they can break down, or what kind of antibiotics they can produce. It's just amazing. So this was our opportunity to start growing them and then keeping them. So you isolate them, and that can be tricky because some organisms have a requirement to grow together. Some are easily contaminated. Some grow like super duper slow that you wouldn't necessarily want to wait for, but we wait, and then we can isolate them, and then we have that collection and we're keeping it for future study, for future biotechnology, for the military. And that takes time and energy, but it's really important because it expands our capability for biotechnology, because you don't want to ignore one particular area just because it's hard. That's where the fun is where it's hard. It's like, oh, this is really cold. It's going to take a long time to grow, but let's do it, because if you're interested in ice inhibition, they've lived with this for a long time. So how can we capitalize on it?
Yeah, sure, sure. That's interesting. Um, Permafrost covers, I think it's like 24% of the Earth's surface. That kind of sounds right from what you've seen, but a whole bunch of the Earth is covered by permafrost. Can you just kind of talk about what the potential for this research can be, kind of maybe where you see the future of some of this research going?
Yeah, so I think it offers an opportunity to expand on what we know about permafrost, how organisms are growing within permafrost itself, or different terrestrial ice. Like you said, it covers a larger portion of the Earth's surface than one might immediately think about, right? And so it's a lot of places, and um we can start, we have started sampling, understanding what's happening as extreme weather conditions, like I said, how cold can they live in, and then also for biotechnology. I've gotten questions of, okay, how do the microorganisms contribute to it staying frozen? Or is there some sort of ice nucleation that can come about to help enable that? And that requires a lot of understanding of the system, tweaking it in the laboratory like we do. And then something critical that ERDC is really good at is taking what we know about the laboratory and saying, let's scale this up. Let's try it in a larger place, right? Like our cold rooms, or our frost effects research lab. Or and then if that works, well then let's make it bigger. Let's put it back out in the field and see if it works, so that we can really see that technology transition fully, and see where there's gaps, and oh, that didn't work out so good. What can we fix to make it an attractive technology?
Great. Well, thanks, Robyn. Thanks for joining us. Very interesting, great discussion as always. So it's great to have you. That's all the time we have for today. But we thank our audience for joining us. We remind everyone you can find more resources about this podcast on our website at poweroferdicpodcast.org. And we'll see you next time.
Great. Thank you, Chris.